Well, thank you, Sam, for your warm words. I would also like to welcome you to this incredible event. Some of you have been at the celebration last night, and I'm sure you appreciated the incredible festivities. But today is really the highlight of this prize ceremony, where you're actually going to hear from the scientists themselves about their work. We live in troubled times in which the truth and the value of rational knowledge is under attack. And I think in that respect, it is particularly important that these events take place. And I would like, as Sam already did, thank the founders of this award for this incredible opportunity to put the spotlight on discovery, on the foundational discoveries that make future process in medicine and in physics possible in this world. And in that respect, I think it's really thank you, thank you, thank you. And that's it. So it's my great pleasure to introduce our first speaker, who is David Julius, my wonderful colleague here at UCSF for many, many years. David started his work in Berkeley, where he worked on yeast, trying to understand the mechanism by which yeast cells talk with one another prior to a mating process. He then went to do postdoctoral work with Richard Axel at Columbia University, where he became a neurobiologist. So you see the transition from a model system into another model system, but a little bit bigger one. And then when he came to UCSF, he started working on the receptors, that mammalian receptors, human receptors, that sense pain, and that sense pain in its various forms, including the hotness of peppers, mustards, spider venoms, heat, cold, and so on and so forth. And for these discoveries of the receptors and how they work, he was awarded this year's Breakthrough Prize in Life Sciences. This work will lead to our increased understanding of how pain is perceived and is transmitted to our brain. And with that, I think can fulfill an enormous and currently largely unmet clinical need. His talk is what's going to happen in the next 10 years. That's the challenge for all of the speakers. Beyond opiates, the future directions in pain therapy. So please join me in welcoming David to the stage. Professor Julius, here you go.
Dr. Walter. All right, after you. Okay, no podium. All right. What do I hide behind? All right, thank you all for being here, and thanks to the Breakthrough Foundation for putting on this wonderful symposium, especially on my home turf at UCSF. It's always great to speak here. As Sam said, it's a little antithetical for experimental scientists to talk about the future, but I think we all must think about that to some extent. And today what I want to do is just talk about what I think are some of the challenges in pain research and where things possibly can go and should go to give us an insight into the future and help patients. So today the title of my talk is Beyond Opiates, Future Directions for Pain Therapies. And the title is based a bit on, of course, the current opioid epidemic, but it really is focused more on understanding how we can treat chronic pain syndromes going into the future, many of which remain refractory to any sort of treatment, pharmacological or otherwise. So one question is, I thought I'd just start off with a very basic question, is why do we experience pain? We usually think of something that has a very negative valence, but we have a pain system for a very important reason, and that's as a warning system to protect us from tissue injury, to signal when there's impending tissue injury or when we need to initiate appropriate protective reflexes. And I would argue, I know I'm biased in this regard, but I think this is our most important sensory system for our survival. And you know this because in people who have either genetic mutations or diseases that render them insensitive to noxious stimuli, pain-producing stimuli, they are at great risk for injury and death, especially young children who haven't learned to avoid things that can hurt them. So we need the system to stay healthy. The problem is that pain can outlive its usefulness as an acute warning system and instead become persistent or chronic and debilitating. And as a result, we're left often with these debilitating pain syndromes, even long after.
You belong on the carpet.
Oh, I have to be on the carpet? Okay. But then the lights are in my eyes. Okay, thank you, Peter. Peter is my senior colleague. He's always steered me in the right direction, so all right. And the challenge, I think, is really to better, one of the challenges, and I'll have 3 or 4 in this short talk, is to better define pain syndromes. And we talk about chronic pain as one thing in the same way that we talk about sometimes cancer, but we know that cancer is a collection of different maladies, and so is pain. And so we need to know how many different types of chronic pain syndromes there are, and really what are their underlying molecular mechanisms, and what distinguishes one from the other if we really want to, at some point, take a more rational approach to treating different types of pain syndromes. And so we know that chronic pain almost certainly has different clinically and mechanistically. This is a collection of clinically and mechanistically distinct syndromes, ranging from things like musculoskeletal pain to migraine to itch to post-herpetic neuralgia, or, for example, the pain that's associated with shingles. And I think one of the future challenges is to understand the signaling systems and the cell types that give rise to these different types of chronic pain syndromes, and the neurotransmitters and other factors that are most associated with each one. And this is important because what it means is that we can hopefully exploit these differences to understand how they can be controlled differentially by differential pharmacology, because we know even now that not one pain drug, even opiates, work to treat every type of chronic pain syndrome. And one of the important challenges, I think, that's already started and has really been going on for the last 10 years, but is a very complex undertaking, and I think still represents a future challenge, is to establish a roadmap. And what I mean by that is to search for molecular and functional specificity along the pain pathway, and to identify, for example, sensory nerve subtypes in the periphery that are involved in detecting noxious stimuli. How many different types are there? What are their different modalities? In other words, do they respond to changes in temperature, pressure, chemical irritants, and how are they discriminated molecularly and cellularly? And that work's been going on for a number of years, but it's actually still not resolved. What are the different target tissues that different sensory neuron subtypes innervate? There are some that, for example, are more prevalent in the GI tract, others that are cutaneous and have different areas of innervation. And that, of course, is going to be a big determinant, for example, in whether you're experiencing a syndrome that affects pruritus, itch, versus irritable bowel syndrome. So we need to know, in other words, what the wiring diagram is and what the specificity of cell types are if we're going to get a really rational mechanistic understanding. And this also pertains to the next way station of information, for example, in the spinal cord, where sensory nerve fibers that detect painful stimuli in the periphery first enter the central nervous system and make their first synapse with ascending spinothalamic tract neurons to take this information to the brain. This is, we know a lot more now about the specificity of sensory neurons that innervate peripheral targets, but we still know very little about who they talk to in the spinal cord and how those signals are sent at that first way station to the brain. So understanding, again, how many types of spinal cord neurons there are, who are their sensory partners, do nerve fibers that detect itch signals versus pressure versus temperature interface with different subtypes of second-order neurons in the dorsal horn? These are still questions that remain to be ascertained, and how do those neurons then connect to the ascending neurons that carry this information to the brain? Of course, pain is not just a peripheral perception problem, but it's in large part a cognitive issue. So the old adage, no pain, no brain, we need to understand how these signals are sent to the brain and what that diagram looks like. And ultimately, we want to know which of these neurons at different stages contribute to persistent pain syndromes, which ones become hyperactivated in the aftermath of injury that lead to a variety of different chronic pain syndromes. And then the ultimate question is, can pain be better controlled? And we have some very good drugs to control pain, such as opiates, non-steroidal anti-inflammatories like aspirin and ibuprofen and acetaminophen. But the problem with these drugs, of course, as we know, is that they have side effects. And opiate receptors are expressed throughout the neuraxis, not only in the periphery, but in the spinal cord and further on in the brain. And the problem is that this means that opiates have a variety of effects, which we are all very familiar with in terms of tolerance, addiction, and constipation, respiratory depression, et cetera. And this is largely a reflection of the fact that the receptors for opiates and opiate-related drugs are expressed not just in the pain pathway, but throughout the nervous system. Similarly, non-steroidal anti-inflammatories and acetaminophen act on enzymes called cyclooxygenases, and they are expressed throughout the body, and this is why these drugs have toxicity, renal toxicity, liver, GI tract, et cetera. So one of the big challenges in this field is to develop analgesics that selectively target pain circuits. And this work has been going on for a number of years, including my own lab, but it's still very much an area that I think will go on into the future for at least another decade, and that is to identify molecular players at the periphery on sensory nerve terminals where there's more of a hope that you can target receptors and other molecules that are more dedicated to the pain pathway. The more peripheral you are before you get into the CNS, namely the spinal cord and the brain, I think the greater chance, or at least this is our hope, that you have to interfere specifically with pain and not with other neurally modulated processes. And the challenge there, which is ongoing and continues, is to identify molecules such as receptors, ion channels, and other signaling molecules in particular that are expressed on the so-called primary afferent nerve terminal, the peripheral nerve terminal that initially detects pain in this process called nociception. And we and others have done that to identify molecules that sort of appreciate things like changes in ambient temperature, in pressure, in chemical irritants, and many people in the field have been working on this for the last decade or so, or 2 decades. And this has given us some insight into not only how we can identify molecules that themselves identify different subtypes within the nervous system, sort of basic neural circuitry issues, but have also revealed potential targets for the development of new analgesic drugs. So we need to continue to identify relevant receptors, and then we need to identify drugs that target these molecules in what I would call in an appropriate way, that act appropriately. And what I mean by that is that the ideal analgesic drug is not a drug that blocks the ability of these receptors and ion channels to participate in acute pain, because you need that acute pain sensation as a protective mechanism, but rather a drug that interferes with the activity of these receptors and molecules in the context of a pathological event, like an injury or post-injury pain hypersensitivity. And this is a real challenge in the field. There are a number of targets that have been identified, such as receptors that my lab's worked on, and there are a number of antagonists or blockers that block these different channels and receptors. And some of them are successful in the clinic, others have side effects, but part of the problem is that you have to develop a drug that specifically can interact with the site on these molecules that we know is involved in appreciating, for example, post-injury inflammatory agents that sensitize these molecules rather than just shutting them down so that you lose the protective function of the pain pathway. And this is really a big challenge in the field for people who do drug development. And one of the ways that we and others are trying to do that is to look at these receptors and channels. For example, I showed two, it's the only data I'll show from my lab here, 2 structures that I've done here with my colleague Yifan Cheng for a receptor for the chili pepper receptor, as you call it, the capsaicin receptor, which also senses heat, and the wasabi receptor, which is a major detector of chemical irritants in our environment. And what these studies are now illuminating are the detailed structure of these channels, so we know not only what they look like and how they're activated under acute settings, but how inflammatory agents interact with these channels to shift their threshold of activation under conditions of injury. And the ultimate drug, we think, in the future would be one that interferes with the modulatory properties of these channels and doesn't just shut them down so that, for example, you lose your protective function for being able to know when you're drinking hot coffee, but instead modulates the activity of the channel when it's activated by or acted on by an inflammatory mediator that shifts its threshold to body temperature so now that you feel pain under conditions where you shouldn't. And so these are the sort of drugs that people are looking for. And I think going into the future, having detailed structural and biophysical information about these molecules will be a way forward to develop drugs that have a more specific action and a better analgesic profile. Similarly, we can exploit computational and natural product chemistry to continue to develop analgesics. This is, again, an ongoing effort, but something that still has a large future to it. And so, for example, there are attempts now by people like Brian Stroikert here and others to develop drugs that act on receptors that might have bifurcating signaling pathways, such as an opiate receptor, and to favor one pathway that's particularly involved in analgesia, and to spare the activation of other pathways that mediate on-target side effects, if you want to put it that way. And this is really an emerging field. It's a very complex one. It's somewhat controversial as to whether you can really pick out a particular intracellular signaling arm of a receptor or channel and have it dedicated to a particular task. But this sort of biased agonism, as we call it, is one area of future research that people are looking at. And the other is really to do what we've been doing over the years and many other people, and to continue to exploit our natural world and let evolution do the work of finding amazing new ligands for channels and receptors that have analgesic properties. And the reason that I think that this is still such a beautiful effect, pathway, is that the main analgesics that we use today, opiate analgesics and nonsteroidal anti-inflammatories, all come from natural products, namely opium poppy and the willow bark. And I think there's still a huge amount of road to be run in this area. And I think it's also an argument for really preserving our environment and our planet, because we still have yet to harvest so much of what nature has provided us in terms of pharmacology. Now, the other thing is that there are also approaches to use, for example, stem cell transplantation. A colleague of mine here, Alan Basbaum, has experimented with putting in human-derived pluripotent stem cells into the spinal cord to replace inhibitory neurons that may be lost during injury, as a way of reestablishing inhibitory tone to reverse hyperexcitability of the nervous system in the context of injury. And this is really sort of a very future area of research. And another one that's used sometimes, and is still a future area of research, is to use neurostimulators to activate or inhibit neurotransmission in the spinal cord under conditions where there's, say, nerve injury that's led to an increase in excitability in the cord. And these are all very exciting and to some degree verified either in preclinical or clinical models approaches to treating chronic pain. But I would say that we still need to understand the basic neurocircuitry, because we need to have evidence-based support for such approaches. This is what, for example, the FDA is looking for in these kinds of mechanisms, where there's either preclinical support or the clinical support is somewhat anecdotal. And we need to really understand exactly what these procedures are doing in terms of the biology of the system, and whether, for example, replacing GABAergic inhibitory neurons in the cord is really a strategically and mechanistically appropriate thing to do under certain cases of nerve injury, such as spinal cord injury or peripheral nerve injury, phantom pain associated with amputation, et cetera. So this is still part and parcel of this very important roadmap going into the future of understanding the wiring diagram, understanding the specificity of neurons within the periphery and the spinal cord that constitute the pathway for pain sensation. And then finally, the question is, one challenge, I think a very big challenge in this field, is can we measure pain objectively? One of the big challenges in the pain field, especially in therapeutics, is that, as we all know, pain is a very subjective sensory phenomenon, much more so than other sensory systems, I would say, such as vision and olfaction. And this leads to a number of problems and challenges in the field. Because pain is subjective and very variable, first of all, it can exist or persist even long after signs of a physical injury have diminished. So when a patient comes in to see a clinician and says, you know, I have pain, it's hard for me to localize it, I don't know exactly where it is. Well, is it related to a certain injury? Well, I think it happened when I was playing basketball and pulled my back or whatever. A lot of times chronic pain syndromes persist long after there's a visible injury, such as in terms of inflammatory bowel syndrome. There may be food poisoning or something that leads to a very fulminant infection that leaves people with hypersensitivity in the gut, but the hypersensitivity persists or in fact sometimes develops long after infection and inflammation have resolved. And so it's very hard to link an injury event to a persistent pain syndrome. And having more objective ways to diagnose and follow this pain would be tremendously helpful to clinicians. And the challenge is to develop tools to assess pain objectively. One area that really has not taken off yet, but is a very big focus for places like the National Institutes of Health, is to develop biomarkers and molecular probes that are associated with chronic pain and hopefully different types of chronic pain syndromes. And the other is to use imagery of pain circuitry to try and objectively see if regions of the nervous system and the spinal cord or brain are activated without having to ask a patient, how do you feel, where do you think your pain is, as a way of objectively measuring this. And so work from people like Irene Tracy have begun to look through the neuraxis and look at patients who are experiencing, you know, mostly acute pain and ask what parts of the central nervous system are activated and what parts of the central nervous system are activated when you impose a noxious stimulus in different ways, be it heat, pressure, chemical irritants, etc. And the question is, can we use these signatures as specific, can we use these imaging patterns as specific images for, to diagnose different types of chronic pain syndromes, to give an objective measure for clinical trials. And one of the big problems in clinical trials for pain is that there's a huge placebo effect, which is itself is interesting. So the placebo effect for most clinical trials hovers around 50%. And most drugs like gabapentin, for example, are just marginally above that placebo level. So can we have a more objective level to, one, ask if a drug is really effective, and two, to understand the nature of the placebo effect more fully, because that's an interesting biofeedback mechanism to control pain. And then finally to use that as an objective measure to see if pain treatment is actually efficacious over the short or long term. And the other thing is that having central patterns of pain that are objective that you could measure would also be very helpful for the design of biofeedback mechanisms and devices that would help us control pain, and where again we could get a more objective measure for whether they're working. But again here I would say that understanding the basic properties of the pain pathway in all these cases is really fundamental to achieving better pain control, whether we're trying to use stem cells, pharmacology, or biofeedback mechanisms. And this way, hopefully, to gain a more precise and effective treatment that, for example, will not be just trying to, you know, use opiates for treating all types of pain, for which opiates are not effective for all types of pain, and understand what makes, for example, headache pain different from GI pain, etc. And finally, I'd just like to thank all the members of my lab over the years who have contributed to the work that we've done, most of which I haven't really talked about specifically, and all our wonderful collaborators here at UCSF and elsewhere. And I'd like to have a particular shout out to the National Institutes of Health, especially the NINDS, which really means you, the taxpayers, for supporting most of the research in my lab, as well as a number of private foundations. And I'd like to thank UCSF and the University of California. It's been my home for graduate work, for postgrad, for all time. I've been a faculty member now. We'll be 30 years here in December, and it's been a wonderful public institution that supported the highest level of research and education and clinical care in the case of UCSF. And thank you to the Breakthrough Prize and the Breakthrough Foundation for hosting all of us today. Thank you.
We have time for one or two questions.
Yeah.
Why don't you pick?
One or two questions, anybody?
Thank you. Would you elaborate on the types of pain, also the syndromes of pain, which are not relieved by opiates. It seems to me that there should be a strong clue there as to what's going on. I mean, what's distinctive about one or the other,
Yeah.
and indicate directions for research. So which ones, what
Well,
are the ones that are not responsive?
One
What does that tell
example
you?
that I can give you that we worked on a model of years ago is pain that results from bone cancer, which is bone cancer pain, which is a breakthrough pain. It's very, it's on a scale of 1 to 10, bone cancer is on 11, or pain is 11 or 12. And that pain is not treated very effectively by opiates or non-steroidal anti-inflammatories. And possibly the reason for that, and may require combination therapy, is that when a tumor is growing in a bone, there's a number of things that are going on to initiate pain. One is there's physical disruption of the bone, which leads to fragmentation of the nerve fibers. So there's an aspect of what we call neuropathic pain, which results from nerve injury directly. And then as the tumor grows, it's spewing out all these inflammatory agents or growth factors that then act on the nerve terminal itself to generate sort of, in essence, a component of inflammatory pain. So it's a complex mixture of both nerve injury pain and inflammatory pain, and so it's probably multimodal in its mechanism. And so one drug that is not likely to diminish that pain, and I think that reflects, a complex situation there where there are multiple points of action on the nerve fiber, both physical and chemical, that are leading to pain, and that probably pertains to a number of instances. And so that's why I say we really need to understand the molecular processes that are occurring in the periphery and the nerve fiber types that are participating in this before we can really sort of take a mechanistic approach to understanding, especially in a complex situation like that where multiple things are happening at once. Yeah.
How do you tackle pain from psychological subject?
Well, that's very, yeah, so the question is how do you, and I should, as a disclaimer here, I'm not a physician, so I don't treat patients, but how do you treat pain from psychological pain? I think that requires interaction with psychiatrists and psychologists, but the question is, the question is a very important one, because that's where it would really be fantastically helpful to have biomarkers and other objective measures of pain. Because pain, when people are dealing with chronic and persistent pain syndromes for weeks, months, or years, it's inevitable that there will be a psychological component, because pain is by its nature a cognitive process. And so I think that we can never divorce that. I mean, I tend to study the more mechanistic aspects of pain because I'm more of a reductionist biologist, and that's, I think there's a very important component there. But I don't want to imply that by understanding all the mechanisms we cannot ignore the psychological and cognitive component of pain. It's so important. But I think having objective measures would enable us to know, is there really a pain, was there an initial event that led to a change in neural function that's associated with a particular type of pain syndrome? Was that what we think now is a psychological component of pain really related to an injury? I mean, I think that's another thing, is that we don't just want to say that people are suffering from pain psychologically, when in fact there might have been an injury that initiated that could lead to a treatment. But I think when people have, chronic pain, especially over years, they need a combination of just not medical intervention, but also therapeutic intervention to help them cope with that from a behavioral level.